Hello, everyone, and welcome to this AnaBios webinar. My name is Chris Mathis, and I'm the Chief Commercial Officer at AnaBios. It's my pleasure to be the host for today's webinar. I'd like to thank our Director of Marketing, Gary Watkins, for his behind-the-scenes efforts to put this together. And I will introduce our speaker in a moment. Before I do that, I would like to let everyone know about a webinar that's coming up in June on a similar topic by Dr. Mike Hildebrand, who's an Associate Professor of Neuroscience at Carleton University. Just came out with a very interesting paper on sex differences. And so make sure you register for this. You can find the link to register on our website. Again, that's June 23rd. And just as today, if the date isn't convenient, you can always register and you'll get a link to watch it on demand at your convenience. So it's my pleasure to introduce our speaker today. Before I do that, I just want to make sure everyone knows about AnaBios. I think many of you in the audience do know about AnaBios, but I'll tell you that we are a unique contract research organization located in San Diego, California. We recover human tissue samples from ethically consented donors and use the tissues or cells to perform physiological assays in which we can test preclinical compounds for drug discovery and preclinical projects. So we have essentially redefined first in human. And so for more information, you can go to our website, anabios.com. And we do have some brochures in the handout section in your GoToWebinar panel related to our DRGs and human spinal cords. So feel free to download those throughout the presentation. And we will have a live question and answer at the end. This webinar is live, by the way, where you can type in your question in the panel, or you can raise your hand and we'll ask you verbally. So without further ado, it's my pleasure to introduce Dr. Sarah Ross. Sarah did her PhD at the University of Michigan in Ann Arbor and a postdoc at Harvard Medical School in Boston in neurobiology with Michael Greenberg. Sarah is now Associate Professor at the University of Pittsburgh Medical School in the Department of Neurobiology. Training the next generation of academic scientists is her personal passion, which is outstanding. And for the last 3 years, she's been collaborating extensively with Alberto Vasquez on a project aimed at understanding the regulation of blood flow. So it's my pleasure to introduce Dr. Sarah Ross and her presentation today on sensory integration in the dorsal horn. Sarah, take it away.
Thank you. It is my pleasure to present some of the work that we've been doing, and thank you for the opportunity to share our research. So I wanted to start by considering the primary afferent landscape. This is how sensory neurons are detecting the sensory environment, and a large number of groups now have done single cell sequencing experiments, and it doesn't matter which of these analyses you look at, because all the groups are converging on more or less the same idea, and that which is illustrated here. This is based on the work by Nikhil Sharma, but it doesn't matter which of these papers you look at, the same idea is kind of emerging. Here you have the A beta fibers on the left-hand side. On the right-hand side are all the C and A delta fibers. And there's a pretty clear idea that there's a bunch of peptidergic fibers that are likely different types of nociceptors. We have thermoceptors, CLTMRs, and then which are important for encoding low threshold input. And then a few populations of primarily cutaneous afferents. Some of these, like the SST and the MRGPRE3 population, are thought to convey itch to the nervous system, whereas the MRGD population, this is largely overlapping with the IB4 population of non-peptidergic afferents, is a population that we believe is normally conveying crude touch to the nervous system, although it has the potential to convey a noxious stimulus, particularly in the context of injury. And if you're interested in that, please look at a recent study from our lab on this topic. So this is the primary afferent landscape. And I hasten to add, this is based on studies that have been done in mouse. There are going to be, and we know there are important differences between mouse and humans. There's a recent paper from Steve Davidson's lab that's really emphasizing these differences. It's going to be important to sort these differences out. But in any case, from the mouse, this is the primary afferent landscape. And for a given region of skin, all of these different subtypes of primary neurons are going to innervate that region of skin, and they're going to convey this information to, at least for the body, to a region of the spinal cord in a fashion that's organized by lamina. So different modalities of information are going to come in at different levels of the spinal cord, with cool information coming at the very top, followed by the peptidergic nociceptors, and then the SST and MRG PRD populations, followed by a variety of different low threshold populations. And so in the in the dorsal ventral axis of the spinal cord, you have different modalities of information that are coming in at different lamina. In the rostral caudal dimensions and in the medial lateral dimensions of the spinal cord, what you have is the somatotopic representation of the body. And so as you move more rostrally, you're representing body parts that are closer to the head. As you move more caudally, you're representing body parts that at least, are closer to the tail in mouse. And medially, you're representing the more distal regions of the body, such as the digits, whereas laterally, you're representing the more proximal regions of the body, such as the thigh. And so in this way, the information that corresponds to a given region of skin is going to be integrated in a spinal microcircuit column. And what I think is one of the big gaps in it is trying to understand what is happening in this spinal microcircuit column. Let's consider a single segment of the spinal cord. There are probably, in the DRG that corresponds to that segment, they're estimated to be about 5,000 A delta and C fibers. And these A delta and C fibers, the information conveyed by these A delta and C fibers is thought to be conveyed from the spinal cord to the brain by way of about 250 spinal output neurons. And so this represents a massive convergence of information, right? A 20-fold reduction in information. And so what I think is a key function of the spinal cord is to convert a rich diversity of somatosensory information into this sparse code. And this is what I would propose is a key function of the spinal interneurons. For this given segment, there are probably about 25,000 spinal interneurons. And this is what I think they are doing. They are converting this diversity of information into a sparse code that is conveyed to the brain. And what our lab was interested in trying to understand is, how is this done? So to tackle this problem, Charles Warwick, who was a postdoc in my lab, decided to take a population imaging approach. And we're doing this with an ex vivo approach in which we have a chunk of spinal cord with the skin and nerves attached. And for these experiments, we put a calcium indicator, GCAMP6, into excitatory neurons by virtue of the VGLUT2 pre-allele. We were also very interested in understanding what is the activity in the spinal output neurons. And so to see those cells in parallel, we also back-labeled those neurons by injecting dye into the parabrachial nucleus. So here is a picture of what it looks like. We're dissecting out a very large piece of spinal cord with the skin and the nerves attached. And this is advantageous both because we get the very, very high imaging quality, partly due to the lack of movement. There's no breathing involved. But one thing we have to recognize is that this is a simplified model. We're not getting the descending modulation that would normally occur in a live animal. And so it represents a simplified circuit. And I think, at least from our perspective, it's good to start with this simplified circuit, but in the long run, we're going to want to layer on the more complex levels of regulation that come from descending modulation. So this is what it looks like from the top down. Right? We are imaging from a region of the spinal cord shown here. And this, if you do a maximum intensity projection after the application of KCl to depolarize all the cells and get a big calcium signal, this is what you see, right? All of these green cells are excitatory interneurons, and the ones that are double labeled with green and yellow, they either look yellow or sometimes they look orange here, are spinal parabrachial projection neurons that are conveying information to the brain, but that also have GCAMP in them. So when we do this preparation, what we have is a situation where we can see some neurons that respond, uniquely to heat, other neurons that are responding to cool, some that respond to brush. And here's an example of a spinal parabrachial output neuron that is polymodal. It's responding to both brush and also cool. Right? And we have very, very good signal to noise, so it's really clear, how the neurons are responding to different stimuli. So these are the different stimuli that we decided to use for our first set of experiments. We used 2 gram and 0.16 gram Von Frey filaments, as well as brush, air puff. Using a Peltier thermode, we did temperature ramps, to either hot temperatures or warm temperatures, cool temperatures, and cold temperatures. And what we see when we look at the diversity of responses is there's all kinds of different responses in different cell types. And so here's an example of 6 different interneurons that are responding in different ways to these stimuli. And here are examples of 2 different spinal parabrachial output neurons that are again responding to these different stimuli. So the first question we had was, how are we going to analyze these data? And so what we decided to do was to term or call neurons responders if they showed a response to the stimulus that was at least 125% delta F over to F, so that the GCAMP signal was at least 125% and 6 times the standard deviation of the baseline period. So we were very confident that we were looking at real signals. And when we looked at whether or not excitatory spinal interneurons were responding to mechanical input, heat and cold, what we found is that although there are some neurons that just respond to a single modality, there really are a lot that are polymodal. And if you look at the spinal parabrachial projection neurons, this degree of polymodality is even more pronounced. Similarly, when we look, this is for the 3 cardinal modalities of mechanical, heat, and cold. We divided the mechanical submodalities, I should say that idea that the spinal parabrachial neurons are even more polymodal is quantified in this graph. We also looked at mechanical submodalities of brush, which is a dynamic stimulus, and low threshold and high threshold punctate Von Frey stimulation. And again, we saw there was a reasonable degree of response of neurons that responded to all 3 mechanical submodalities. And this type of pan-mechanical response was, again, even more pronounced when we focused exclusively on the spinal parabrachial output neurons. And so this kind of really left us in a tricky place where a lot of neurons are responding to many, many different types of stimuli, and we wanted a more satisfying way of thinking about these data. And so at this point, we decided to switch to thinking about things in terms of their tuning. And so the idea is that although a neuron might respond to a bunch of different types of stimuli, they're nevertheless tuned to one stimulus type over another. So let me walk you through the way that we decided to do this. We decided to present the data in terms of vectors. Here's an example of a cell that's responding only to brush, not to cold or heat. And so when we add the 3 vectors, there's almost no heat, almost no cold, a large response to mechanical, and the sum vector is shown here in black. Here's an example of a different cell. This cell is responding a little bit to brush, not really to cold, lots to heat. So here is lots to heat, a little bit to brush, not to cold. When we sum the 3 vectors, we get a sense of the cell's tuning. This is a mechano-heat cell, but it is tuned for heat. And finally, here's an example of a cell that really is responding, quite robustly to brush, cold, and heat. when we sum those three vectors, you get this very, very stubby black vector shown here. And so this is what it looks like if you have a truly polymodal neuron that does not show tuning. We can use a similar, so when we look across the cardinal modalities, what we see is that in the superficial dorsal horn of the spinal cord, and I should emphasize, we were imaging neurons in the first 60 microns of the dorsal horn. And so this is like lamina 1 and lamina 2. So across neurons in lamina 1 and lamina 2, we see neurons that span the gamut in terms of their tuning for different types of sensory stimuli. And we can apply the same type of analysis looking at the mechanical submodalities. And again, the neurons are kind of spanning the gamut in terms of their tuning. So these data suggested that although there's a lot of polymodal neurons in the dorsal horn, they're nonetheless tuned to detect different types of stimuli. Another thing that we wanted to look at was the somatotopy, because we know that the dorsal horn is somatotopically organized. And so to do this, we poked different regions of skin with the von Frey filament. And what we found, which is consistent with understanding of how the dorsal horn is organized, is that when we poke more proximal regions of skin, shown here in red, it activates neurons that are located more laterally in the spinal cord, whereas when we poke more distal regions of skin, as shown here in blue, we activate neurons that are located more medially in the spinal cord. Here you can see the traces from individual cells, and these ones in the middle are responding to both stimulation in two different sections of skin. And so this is a way of looking at somatotopy that is flipped from what people normally do, right? People are normally recording from a single cell and poking different regions of skin. Here we were poking a single region of skin and asking, what are all the cells that respond? And then we did that over various regions of skin. And in this way, we showed, very clear somatotopic organization in the dorsal horn, consistent with our understanding of how the spinal cord is organized. So this is satisfying. We can characterize the tuning characteristics of neurons, and we can see the somatotopic organization. The problem that we still had was that we're still looking at a whole bunch of excitatory interneurons for the most part, and we can't tell which kind are which kind. And so how are we going to model how these different cell types are integrating information, right? We know that there's lots of different excitatory interneuron subtypes, and how are we going to distinguish them if they all have GCAM? So what Charlie discovered was this, and developed, was this novel approach to classify different cell types within our preparation using an approach called cicada, or cell type identification by calcium coupled activity through drug activation. And the basis of the idea is that different neuronal subtypes might express different GQ coupled GPCRs. And because GQ coupled GPCRs are coupled to the release, IP3 mediated release of calcium from internal stores, this would allow us to see which cells expressed which GPCRs. And so what we did in these experiments was we performed the whole experiment, and then at the very, very end, we applied TTX, which blocks action potentials. So now if we see a calcium response, we know it's due to the activation of these receptors. And then we applied a bunch of excitatory GPCR agonists in series, spaced 5 minutes apart. Right? So we applied all of these different GPCR agonists with the goal of visualizing the neurons that expressed each of these different receptor types. And here's an example of the calcium kinds of calcium traces we're getting. It's a really huge, easy to detect delta F over F signal, where we can say with reasonable confidence, right, these are neurons that express the receptor for CCK. These are neurons that express the receptor for GRP, etc. Of course, it didn't have to be, you could have a neuron that was expressing receptors for all kinds of agonists. And so at first, we weren't sure the best way to classify this. To do it in an unbiased way, we decided to do dimensionality reduction. through clustering. And what we found was that these clustering programs suggested that there were 7 types of excitatory neurons in the dorsal horn. Let me walk you through what those look like. Okay. So here is an example of an experiment. It's in the presence of TTX, so all this calcium release is due to direct action of these agonists. We're applying in series, GRP, TAC1, CCK, etc. And what you're looking at is for each of the, each of the clusters. In the talk, I'm probably going to call these cell types, but of course we don't know their cell types. What we know is that they are, groups of neurons that are responding similarly, have a similar subset of GQ-coupled GPCRs. Sorry about that. It turned out that many of these different excitatory clusters could be defined based on their response to a single agonist. So take example, excitatory 2. This is a population that is responding pretty much exclusively to TAC1, which is substance P. Here we're looking at the median plus or minus the interquartile range for this population of neurons. Here's another example. These cells here in excitatory 4 responded to CCK, but not to any of the other agonists. So it looked like most of these excitatory neurons expressed, a single type, or predominantly a single type of excitatory GPCR, but that wasn't always the case. Here's another example. Excitatory 1 is a population that's responding both to TAC1, which is substance P, and also to TAC2. So this is another way of looking at these responses in this polar plot, where you can see that, for instance, excitatory 1 responds both to TAC2 and TAC1, whereas the CCK population, excitatory 5 rather, is only responding to CCK. Here you can see the distribution of these cell types in the dorsal horn. I hasten to add, this is the distribution of neurons that responded to one of the cicada ligands. There were some neurons that didn't respond to any of these ligands, right? So this allowed us to see 40% of neurons in lamina 1 and about 25% of neurons in lamina 2 because they had these receptors. There's still lots of excitatory interneurons that don't have receptors for any of these ligands, and we can't, further classify them at this point. And I'll also add, we screened more ligands than this. We screened ligands for which there was any reason to think that there might be receptors in the dorsal horn, and they were known to be GQ-coupled GPCRs. So we think we've, captured the vast majority of ones we're going to be able to see based on what we know now. Of course, there's a bunch of orphan GPCRs that we don't yet have a ligand. Those, when they're discovered in the future, we're going to be able to layer on more detail. And we're hoping we can extend this approach in the long run to look at GS-coupled GPCRs and maybe GI-coupled GPCRs to give us even further detail. Nevertheless, this is allowing us to see many different neuronal populations in the dorsal horn. And when you look at the depth of these different putative cell types, you see that excitatory 1, 2, 3, and 4 are found in lamina 1, whereas excitatory 5, 6, and 7 are predominantly in lamina 2, consistent with the idea that these really do represent, different cell types. So now we have the ability to characterize the tuning of different neurons to different somatosensory stimuli. We can see the somatotopy, and we can further classify cells based on their cicada profile. We call these cicada-defined populations. So then we were at a stage where we wondered, what questions should we look at in the dorsal horn? And we decided that as a first question to tackle, we would look at the question of, what is at a population level, what's happening when in the context of capsaicin-mediated central sensitization. And the reason that we picked central sensitization as our first model to look at is because central sensitization is a very well-characterized phenomenon, both in human psychophysical studies and in all kinds of mechanistic studies. When you inject capsaicin into the skin, it causes for a few minutes a sharp burning pain. This is the primary response. And then that pain subsides, but it is followed by a prolonged period of mechanical allodynia that lasts for hours. And this mechanical allodynia is known to be due to plasticity at the level of the dorsal horn involving central sensitization. This has been characterized in detail, one cell at a time. We thought this is a fantastic opportunity to see central sensitization at the population level for the first time. So that's what we did. And we probed the area. We injected a small amount of capsaicin into the skin and then did experiments to look at mechanical allodynia. So when you inject capsaicin into the skin, what you see is that there are a bunch of neurons in the dorsal horn of the spinal cord that respond immediately. This is these are the raw traces. This is what it looks like in a heat map. And for, if you inject 10 microliters of capsaicin, what you see is there are about 40 neurons in the dorsal horn that are responding with a time course that matches the psychophysical experience of intense burning pain for several minutes. And so we think these neurons are involved in processing that initial input and conveying it to the brain. Looking down through the microscope at which cells are activated, we can see that they're clustered in a region corresponding presumably to that region of skin. And shown in these triangles are the projection neurons. These colored in triangles here are the two projection neurons that responded immediately to the injection of capsaicin into the skin. So now we can see at a population level. These are all the neurons in the spinal cord that are responding to capsaicin. What are they? And what we discovered was that the capsaicin responding neurons were primarily this excitatory 1 population. 42% of the neurons that responded directly to this capsaicin application in the spinal cord were in this excitatory 1 population, which are neurons that express the receptors for TACR3 and TACR1. And when we look at the characteristics of this population, this is a highly polymodal type of interneuron that is responding to all kinds of input. So, so these are the neurons that are the interneurons that are responding, immediately when you inject capsaicin. The real question that we were interested in was when you inject capsaicin into the skin, what's causing this this allodynia? And so to address this question, we looked at the response properties of neurons either before or after the injection of capsaicin, and then we applied cicada at the end of the experiment. First, I want to show you at a population level what allodynia looks like. So if we were to apply a von Frey filament to a given region of skin under basal conditions, here you see a small number of neurons that are responding. And when you poke other receptive fields, you don't see responses. Following the injection of capsaicin, we see a couple of different things. One thing is that there's many, many more neurons that are responding. They're responding with a greater amplitude of response, and also you can activate these neurons when you poke many different areas of skin, including those, from either receptive field 1, 2, or 3, you're still activating neurons in this field of view. So these are the same data plotted, a different way, where it really was, in gray, you're seeing the response properties of all of the excitatory neurons at once, the whole population, either under basal conditions or following the application of capsaicin. And what you see is that there really is an amplified input that is selective to, low threshold. This low threshold that didn't normally activate very many cells now is activating many, many more cells. And so we think this corresponds to the psychophysical phenomenon of allodynia. Here's another way of showing those data where you're just looking at the change. The change really is to the low threshold input. The other thing that we see is that more cells are responding and the receptive field sizes are significantly increased. And another way of looking at the data is thinking about the tuning. So here is an example of looking at the tuning of the population, which is shifting after capsaicin treatment towards low threshold stimuli. Our real question was, I mean, this is what it looks like at a population level. It's consistent with the phenomenon of allodynia, and it's consistent with the phenomenon, I should have emphasized this, that when you inject capsaicin into the skin, you're going to feel burning pain in that very spot where the capsaicin was injected. And then you're going to feel allodynia over a much, much broader area of skin. And that kind of extended area of allodynia might be due to this increased receptive field size. The question we were really interested in was, which neurons are actually mediating this? And so to look at this, we looked at the different cicada populations. And what we found was that there were 3 different populations, excitatory 6, excitatory 5, and excitatory 2, that changed significantly after capsaicin treatment. So let's look at these 3 populations in turn. Here is excitatory 6. This is a population that is in lamina 2. Before capsaicin treatment, this is a population that wasn't that responsive to any different types of stimuli. After capsaicin treatment, this population is responding much more robustly to lots of different kinds of input. And so we think of this as a population that's generally amplifying input following capsaicin treatment. When we look at the tuning, in contrast, there's no significant change in the tuning. However, when we look at the receptive field size, in this population, there is a huge increase in the receptive field size. And so this population could be contributing to the cyclophysical phenomenon that the area of allodynia is over a much greater area of skin than the initial capsaicin injection. We saw a different picture when we looked at the excitatory 5 population. This is a second population that is in lamina 2. Here we found a population that really selectively showed changes just to low threshold input. You can see that here a different way where there's a significant change in its tuning towards low threshold input. However, its receptive field size did not change at all. So now we're looking at 2 different populations. One that is showing this shift to low threshold input, that could be the allodynia. Another one that's showing this increase in receptive field size. We're thinking of that as the, potentially mediating the large area of central sensitization. Both of these are in lamina 2. We believe the key way this information is getting to the brain is via lamina 1. So we were interested to see that the third population that was changing significantly was a population in lamina 1. It's excitatory 2. And what we saw in this population was kind of a combination of both changes. There's a significant change towards low threshold input, as well as a significant increase in the receptive field size. And so it's early stages yet. We don't have, the connectivity map to actually, know this is how the neurons are connected. But, this is at least suggesting a model that when you inject capsaicin into the skin, it activates a population of TRPV1 expressing primary afferents that activates excitatory 1 for 3 to 5 minutes, and that the activity in excitatory 1 is driving changes. We don't exactly know the nature of these changes, but driving changes in plastic changes in the spinal cord that are that mediate the allodynia, involving excitatory 2, excitatory 5, and excitatory 6. We're thinking of excitatory 5 as being a population that's shifting its tuning towards low threshold input and potentially meeting allodynia. We're thinking of excitatory 6 as an amplifier that's showing a great change in its receptive field size. And maybe excitatory 2 is integrating both of these inputs in lamina 1 before it's being conveyed to the brain. The other big question is, how is this information being conveyed to the brain? And so far, I haven't told you very much about what's happening in the spinal projection neurons. That's what I want to talk about now. So when we looked at how the activity in spinal projection neurons changed following treatment with capsaicin, what we found was that they too showed a significant shift in their tuning towards low threshold input. We decided to classify these based on ways that these cell types have been classified historically, which was, wide dynamic range, low threshold, and high threshold. And what we found is we have, some of each of these types of spinal output neurons. And when we look at the ones that are showing this type of change in their tuning, it turned out to predominantly be this wide dynamic range population. It's showing the greatest shift in tuning. There's no significant change in the low threshold spinal output neurons and a small change also in the high threshold neurons. We saw the biggest change with these wide dynamic range cells, and we thought it was extremely interesting that these wide dynamic range spinal parabrachial output neurons were the subset that responded most to these peptidergic agonists. So this was the population that was responding to TAP1 and NMB and TAP2 and other peptides, whereas the high threshold spinal output neurons and the low threshold spinal output neurons were much less likely to respond to any of these GPCR agonists. And so when we put this all together, what this is suggesting is that the excitatory input from the TRPV1 afferents is acting on excitatory 1, which is changing the properties of excitatory 2, 5, and 6. We don't know this, but we like the idea that information that's changed in excitatory 5 and 6 is being integrated by excitatory 2 and eventually conveyed from the spinal cord to the brain, possibly via a population of wide dynamic range in combination with high threshold spinal parabrachial neurons. And so this is really our first step in trying to understand how it is that the 25,000 spinal interneurons for a given segment of skin are working together to integrate this complex input so that it can be conveyed to the brain through a sparse code. And what we're doing as our next step, here I showed you a kind of a picture of what's happening in excitatory neurons. What we're doing next is we're imaging from all neurons, both excitatory and inhibitory, but we can tell which ones are which because the inhibitory ones have nuclear M cherry in them. And this allows us to see the activity across all neurons at once, including the spinal output neurons. Plus we're layering on cicada to see the putative cell types. And we think this is gonna kind of allow us and other groups who want to do similar kinds of things to do a new level of modeling of how somatosensory integration occurs in the dorsal spinal cord. So with that, I want to acknowledge the team of people that was involved in doing this. Charles Warwick, who's shown here, is the postdoc who really spearheaded this project and did the imaging and analysis that I showed you today. And there's now other people who are really excited about doing this too and have joined him on the project, including Taylor and Kelly. We are a dynamic team at the University of Pittsburgh and part of the Pittsburgh Center for Pain Research. We are always looking for people who are interested in doing this kind of research and feel passionate about understanding, how pain is conveyed from the periphery to the brain. And if this is something that you're interested in working on, please reach out to me. We'd love to collaborate. We're always looking for new trainees. I'd be delighted if you want to reach out to me. My contact information is here. And with that, I will pause and take any questions. Thank you.
Well, Sarah, thank you so much for the excellent presentation. We do have plenty of time for questions. So those of you in the audience, you can write your questions in the question box in the GoToWebinar panel. Or you can raise your virtual hand and I'll unmute you if you would just introduce yourself when you ask the question. And while those questions are coming in, remember you can download the AnaBios DRG and spinal cord, human DRG, human spinal cord brochures also in that panel. So I'll get things started, Sarah, with kind of a general CNS question. I know there are interneurons in the brain also. Is the processing in the brain similar to what you've described in the spinal cord?
So you're talking about the processing of pain, is that correct?
Just in general, or are there other examples of this type of interneuron processing that are similar?
I feel like as, the neuroscience community is just starting to figure out, how different regions of the brain are integrating different kinds of information. And so we don't really know the answer yet. But my feeling about it is that there are regions of the nervous system that are have very specialized cell types, and that kind of from the get-go are supposed to wire a certain way, that are important for fundamental physiological functions like, being able to walk, breathing, everything that's done by the spinal cord and the brainstem is kind of hardwired to almost work from the get-go. Whereas there's other parts of the brain like the cortex and the cerebellum whose function is to learn stuff. And those are going to be two fundamentally different kinds of circuits. In the cortex and in the cerebellum, nothing is going to be represented until the organism tries to learn something. And then what that represents in different animals will depend a lot on what that animal is learning. Right. Whereas the circuitry in the spinal cord and brainstem and midbrain is, hardwired is the wrong word, because of course there's lots of room for plasticity, but it was during development specified to wire a certain way and function a certain way. And I think in those regions of the nervous system, there's going to be a greater diversity of cell types. I think there's going to be a huge diversity of cell types. Almost all those regions have both excitatory and inhibitory interneurons that are going to be working together. And we don't really understand if you think about what we do understand, when a primary afferent fires, it's sending an excitatory signal, giving rise to the release of glutamate into the dorsal horn. And we think we can look at the activity in that primary afferent and understand what it is representing. Once you get beyond that, the information that's encoded in the neurons becomes much more abstract. And I think we're just scratching the surface in terms of understanding how that abstract information is encoded and how the nervous system uses that information to make decisions.
Thank you. Yeah, and this is a great start for neuroscientists. So we're getting some questions from the audience. I'll read them to you. So Sadet Inan said, "Great results, Dr. Ross." 2 questions. One is, have you seen any sex difference? And second, if you had given capsaicin into the paw and then did the same experiment with paw skin, would you have similar results? Could you comment on that?
Yeah, those are great questions. So we did our experiments using tissue from both male and female mice, and we did not see any obvious differences, but we did not power our experiment to specifically look at whether there were differences between males and females. Every time we do experiments, we're collecting more and more data, and at some point we could go back and split it into data from males and data from females and analyze it with that question in mind. And we haven't done that yet, but I think it's going to be important to do. My instinct is that, well, I guess we'll just have to see. I don't know. We'll have to do that experiment. There was not an obvious difference between males and females, but we might see differences if we look more carefully. The second question
about if you give capsaicin into the paw and do the same experiment with paw skin.
So I'm sure there would be some differences, because the innervation of the paw, which is glabrous skin, is a little bit different than the innervation of the hairy skin. All our experiments were done on the hairy skin. So there are absolutely going to be some differences, but I think the kind of major take-home message would probably be similar. And so at the very beginning of the presentation, I had this kind of cartoon of a column microcircuit, and that's going to be a repeated unit going up and down the spinal cord. It's going to be subtly different at different levels because of the different input that's coming in. And so at a level like L4 and L5, where there's a higher proportion of cutaneous input from glabrous skin, those microcircuits are going to look a little bit different than a region of the body that has a much higher ratio of like visceral input. But I think it's going to be kind of a different ratio with different interneuron types and subtle changes in the way that information is processed, as opposed to completely different cell types and a different type of organization. But again, I'm speculating. We will have to do more of this kind of experiment to really test that idea.
Okay, thank you. Here's a question from Amanda Klein at University of Minnesota. She said, great talk, Sarah. Were the data from the GCamp mice recorded from stimulation on hairy or globoria skin? I think you just answered that. If you haven't done both, do you predict there would be a difference in tuning? Does your data give any indication when neuronal clusters are involved in secondary hyperalgesia after capsaicin injection?
Yeah, so I mean, Amanda's question, that's great. It's very similar to the first question. From the globular skin, you're going to have a more specialized cell types like Merkel cells, because that's where there's a higher density of primary afferent input, and there's especially a higher density of afferents that are like A beta fibers that terminate, associated with specialized nerve endings. And so for that reason, the input's going to be a little bit different, especially to deeper lamina. Keep in mind, though, that for these experiments, because it was our first experiment, we just imaged the first 60 microns in the spinal cord. We're only looking at lamina 1 and lamina 2. So for lamina 1 and lamina 2, we're just looking at, C fiber input. I'm not sure that it would be that different, honestly, but we don't know because we haven't done the experiment. When we were doing these experiments, we can, to get a reasonably good frame rate, we can record from a certain, area at once. And we recorded through kind of like 3 different planes to sample neurons in different lamina, over a given area. And if we wanted to, we could double the number of planes to go deeper, but do it over a smaller area of dorsal horn, and collect data from the same number of neurons, but that we're representing neurons that go deeper. We can see much deeper right into lamina 3 if we want to. And so for future experiments, we might look at a different, aspect of the integration and focus on neurons that are, in deeper lamina. That's, still in the next steps.
Right. Yeah, I'm wondering if you could comment on the technology you used for the calcium imaging, and especially in the spinal cord.
Yeah, so we thought about this a lot, like what's the best way to do this?
Mm-hmm.
And in the end, I mean, there's so many attractive reasons to do experiments in vivo. But in the end, at least for our group, we decided that we were going to get a lot more power by doing this using the ex vivo preparation, even though we know this is like a simplified model. But my instinct about it is, we know so little about what these circuits are doing. It's okay if it's slightly a simplified model. Once we have a better handle on what's happening, even within the simplified model, then we can layer on the descending modulation. And I think the descending modulation is going to be so important. But I think it's also okay to ignore it for now. What we like about this preparation is really good signal to noise, especially because there's no movement artifacts because the animal's not breathing. Also, the animal is not under any anesthetic, right? This is an anesthetic-free preparation because the animal's dead. The other, I think, huge advantage to this is it's really amenable to the pharmacological analysis of different cell types, which I think gives us so much more power in terms of understanding the integration. And I think that would be very challenging to do in vivo. So for all of those reasons, we're favoring this ex vivo approach for now, with the view of moving, in vivo at the right time. And that's going to be really important too. In terms of like technically how we do these experiments, we have a Cree allele that's driving expression of GCamp from a genetic allele, like a ROSA locus-based Cree-dependent GCamp expression. We like that because it gives, not crazy high levels of GCamp, but plenty for us to see the signal. And we started doing our experiments with GCamp 6S. Now we're switching to faster GCamps and later GCamps like GCamp 7 and towards GCamp 8. I don't think it's going to make that much difference, but we're able to use the latest GCamp as we can switch. And then we are doing our imaging with a 2-photon microscope. Okay. And as I mentioned, that for this first experiment, we were imaging in 3 planes. We could be imaging in more than we can image in more planes, but just to have the right to be able to image at a high frequency. If we do more planes, then we have to do fewer a smaller area for each plane.
Okay, thank you. Here's another question from the audience. Can you speak to the importance of primary sensory afferent sensitization in the circuit? For example, could you use this imaging technique to understand how afferents might be tuning their modality?
Yeah, so in this particular experiment, we only imaged from the superficial dorsal horn, but it would be very, very easy to just shift the preparation over a tiny bit and be imaging from the DRG neurons instead. And so we absolutely could be characterizing, how primary afferent activity is changing in response to injury or inflammation or something like that. We didn't choose to do that in this experiment, but we absolutely could. And we don't have the ability to see simultaneously the activity in the dorsal horn of the spinal cord and the DRG neurons because they're physically far away enough in space that we can't see them both at the same time with our objective, but they're too close to have two different objectives. So, but one day, figuring out a way to see both those simultaneously, I think, will be really, really interesting.
Okay, thank you. Someone in the audience wants to know if your group is going to look at other pain models in the mouse imaging system, and if so, which ones are most relevant for translation to human pain populations? For example, diabetic neuropathy, chemotherapy, herpes zoster, surgical models of neuropathic pain.
Right. I love that question. Well, maybe not surprisingly, given my lab's history, but we're really interested in itch. So we've started doing some experiments looking at itch. And then when it comes to what's the most relevant pain model, I think the answer is we're going to have to do a bunch of them. And I think what we're going to see, and this would be totally consistent with a lot of work from Becky Seal's lab and others, is that different populations are responding differently depending on the kind of injury. And so if you have ongoing inflammation, you're going to see something different in the spinal cord than if you have ongoing neuropathic pain. And
right.
Seeing that at a population level and understanding those differences is going to be really, really important. And the other thing that we haven't started to look at, but I can't wait to do, is when you apply these analgesics that are blocking pain. I actually think this is relevant for pharmaceutical companies. When you have a novel analgesic that you think might be a good pain treatment, really understanding if it works at the level of the spinal cord or below, we should be able to see it working in this preparation. And you not only see that it is working by virtue of seeing decreased output from spinal projection neurons, but also understanding how is it working, which interneuron populations is it targeting, or primary afferent populations is it targeting to mediate its effects. And I think as costs so much money to take a new drug all the way through clinical trials, I think it makes sense to spend a little bit of time really understanding how it works at the circuit level, potentially as an intermediate step as you're going in the direction of clinical trials.
Absolutely. And I think the key issue there is translatability, as the last question hinted at. At the beginning of your presentation, you mentioned the primary afferent landscape and Steve Davidson's paper that shows there's important difference between mice and human. Can you speculate, or do you have any idea how some of the differences would be if you were able to do this in human spinal cords, for example?
I love the idea of being able to try some of these experiments in other species, including human spinal cord. And there's no the great thing about cicada is that there's no reason why cicada wouldn't work in human spinal cord.
Okay.
And I think instead of putting in a genetically encoded calcium sensor, we could bath apply something like Pura to do calcium imaging in spinal cord neurons. And so there's absolutely no reason that we couldn't be doing almost, exactly this kind of experiment, except that you don't have the skin attached, but some of the similar kinds of experiments from spinal cord tissue, and looking at the question of, conservation, and also the question of translatability. And it's going to be critical to do that, because although I think, it's clear just based on the laminar structure of human spinal cord, and across all species, right? There's going to be elements of in terms of how the circuitry work that are conserved, but there's going to be really important differences too that are probably a little bit more subtle, but are going to be key differences that we need to understand as we're trying to move from an animal model to a therapeutic treatment in humans.
Right. Well, we'll have to follow up with you on that at AnaBios. So we're getting some more questions. We'll do a few more. have you observed any difference in the circuitry between lower lumbar and upper thoracic levels, for example?
Just because of the ease of doing it, we started in lumbar levels, and that's because the spinal cord, as you know, all the fibers that are ascending fibers that go to the brain kind of cover the spinal cord, and the white matter gets thicker and thicker as you go up. And it's easier to see through that white matter in, regions that are more caudal. And so we started our, plus we already had kind of worked at the details for how to do the skin nerve spinal cord preparation, for the hindpaw skin. And so that is what we started with, and we haven't tried anything else. There's no reason why it would be harder, but there's no reason why you couldn't do something similar at a cervical level. There's going to be a few more challenges when it comes to the imaging, but I think they're surmountable, and it would be important to do those comparisons, and we haven't done them at all.
Okay. Have you tested the effect of opioids on this circuitry?
We're so interested in that. I'm personally really interested in kappa opioids because kappa opioids inhibit itch. I'm also really interested in mu as a really potent and powerful painkiller. When the dogma in the field is that mu is inhibiting the release of from the primary afferents, and it is definitely doing that. But there are lots of spinal cord interneurons and spinal projection neurons that also express the mu opioid receptor, and we're going to be able to understand which those cells are, and see how the presence of morphine is affecting the way that the information is being processed through these circuits, I think at a new level of detail. And that's like one of the next experiments we want to do.
Great, can't wait. All right, we'll do two more questions so you don't lose your voice here. One is, could imaging the inhibitory interneurons in the dorsal horn help uncover the mechanisms of central sensitization observed in this capsaicin model?
I love that question. So there's a bunch of question marks here, right? So let's just think about how is the information from excitatory 1, changing the properties of excitatory, 2, 5, and 6. And one possibility, and this is attractive, is that excitatory 1 is like releasing a neuropeptide or something that's having a kind of longer lasting effect. And another possibility is that, excitatory activity in excitatory 1 is like changing the way that the inhibitory interneurons, are working, and we're losing some inhibition. And that's why we're seeing this exaggerated response after allodynia, is because the inhibitory neurons are, not engaged the same way. And the preparation that I've shown you today, we can't see at all what the activity is in the inhibitory neurons, but that's really the next step of what we're trying to do, is look at excitatory and inhibitory neurons in parallel. And we anticipate that there's going to be models of injury where there is, less activity in inhibitory neurons, and we're going to be able to see that. And we're also going to be able to characterize, what kinds of inhibitory neurons using the same cicada-based approach. And I think that's going to give us, a lot, it's going to be different depending on the model of injury. But I think that's going to give us a lot of new insight in terms of what are the best targets if you want to reverse that kind of pain.
Excellent. And finally, someone in the audience wants to know if these data have been published. If so, how can we get our hands on that paper or papers?
So we submitted the paper, it's under review, and we uploaded it to bioRxiv. And so you're welcome to read the paper, or at least our first version of the paper, at bioRxiv. And if you find anything wrong with it, please tell us, because it's under review and we can fix it.
All right, that's great.
So yeah.
Awesome. Well, we look forward to seeing more of what's coming out of your lab in the future. Sarah, thank you so much for this excellent presentation. A lot of great questions. Again, I'd like to thank Gary Watkins, our Director of Marketing at AnaBios, for putting this together. I'd like to thank the audience for the great questions. And be sure to register for Mike Hildebrand's presentation in June on a similar topic with human spinal cords in this case. And so again, Sarah, thank you so much. And thanks everyone for attending.
Thank you.
Bye.